to get these questions addressed. As you have heard, we are recording this meeting. It will be available afterwards on our YouTube. We will share it after the presentation has been totally recorded. So I think while we are waiting for some people still to enter, I think we can slowly start, and I will present the first speaker of today will be Professor Laurens Pauwels. He's currently leading the Laboratory of Plant Transformation and Gene Editing at Ghent University, Department of Biotechnology. He did his PhD with Professor Alain Roses, also from Ghent University, where he contributed to elucidating actually the jasmonate pathway in Arabidopsis. He went to UC Davis with Judy Coles in 2010. They're focusing more on protein ubiquitination and degradation. And when he finally returned back to Belgium, he established some CRISPR-Cas, or some genome editing protocols for different organisms, including yeast, Arabidopsis, and tomato. And this led him in 2017 to establish the crop genome engineering facility at the BIBU Ghent Center for Plant System Biology. And thus he was able to provide services to academic groups, but also to biotech companies, next to improving methods for maize, wheat, sorghum, soybean. And since September 2024, he has become professor at Ghent University of the Faculty of Bioscience Engineering, and where he's also teaching plant biotechnology. His current new research group focuses on advancing plant transformation and CRISPR-Cas9 gene editing technologies in various crops, including engineering agrobacterium strains and integrating haploid induction with genome editing. With these little introductory words, I would propose Professor Pauwels to share his slides. Yeah, thank you very much.
Okay, thank you, Mark. Thank you for quite an extensive introduction. And so also many thanks for your invitation to present at this online cafe. So it's I think a very nice format to meet other people also interested in plant biotechnology. So today my title of my presentation is indeed Agrobacterium-mediated transformation of cereals for Striga-resistance. So my talk will mostly focus on the technical aspects of transformation of plants, and that will be an introduction, I think, to the work of Stephen Runo later. So a bit of an outline. I will discuss a bit of the interest in sorghum and the transformation bottleneck that is in general present in cereals. And then I will talk about three aspects that has led to significant improvement in the transformation of cereals in general the last couple of years. And the first subject I will discuss is improving agrobacterium-mediated transformation using ternary vectors. Then the use of morphogenic regulators to enhance regeneration. And then finally discuss alternative explants for transformation and the use of leaf fragments. And finally, I will discuss a little bit how this fits in the context of the crop genome engineering facility at VIB, and then indeed also introduce my new lab at Ghent University, also working on these topics. So first, maybe sorghum. So Striga, which is a topic of today, is actually a problem for many cereals, but I think especially for sorghum, this is a major problem. Now, sorghum in general is getting more and more attention, also here in Europe, as an alternative crop. Many varieties are naturally already drought tolerant, and so in the context of climate change, of course, people are also looking into sorghum as an emerging crop here in Europe. But nevertheless, research in sorghum, and especially if we're talking about breeding using these new genomic techniques like CRISPR-Cas9, we see that the transformation of sorghum is still a bottleneck. Now, luckily, also for maize, this is a bottleneck, but we have seen that over the last years there was a tremendous improvement in maize transformation technologies, and we see that these technologies are now also leading to improved sorghum transformation, and that's a bit what my talk will be about. So if we look at a typical transformation process, and so this is now for maize, but it looks very similar for sorghum, is that you have these different steps where you start from a certain explant. Typically for cereals, these are immature embryos. These are then co-cultivated with Agrobacterium. And so Agrobacterium is still the main method by which plants are transformed, also for gene editing. And then this is then followed by many steps of tissue culture where transgenic cells are selected and then regenerated into plantlets, transgenic or gene-edited plantlets. And here, with this regeneration step, we will see that morphogenic regulators will help us with it. And then finally, of course, you have this quality control where from all these plants that you regenerate, you have to find then the best transgenic or gene-edited events. So if we look at gene editing, in particular, if we look at the typical pipeline of gene editing in plants, you have indeed these many steps that are involved, going from construct design to finally phenotyping of your plants in the greenhouse or the field. But today I will focus then on this part of transformation. Here on the right, you see, of course, this is from a recent publication we published last year. This is a very complex method with many steps over a long period. I will not go into detail today in all of these. And in general, it's a very laborious method, and also the efficiency can be, as you will see, either low or variable. And so improvements are certainly welcome there. And so the first advancement I wanted to highlight today is these ternary vector systems. So what do we mean by that? So if you look at the wild type, the original Agrobacterium strains, they have a TI plasmid, a tumor-inducing plasmid, and where you have your virulence genes, and you also have a natural tDNA that will be transported to the plant cell, will integrate in the genome. Now, already very early in the 80s, people have developed what we call binary vectors. These vectors, they hold the tDNA, and then the TI plasmid itself has been what we call disarmed, and so the normal tDNA has been removed. So this has been used, of course, and still being used as the standard way of working with Agrobacterium. But recently, people have introduced what we call ternary vectors. These are vectors, a third vector that we introduce in Agrobacterium that now contains an additional set of these virulence genes, these vir genes that can be from the same Agrobacterium strain or a different strain to help the virulence. So there are several companies, several academic groups that have developed ternary vectors. And we ourselves, tested if a vector published by Zhang et al. In 2019, if we could use that in our maize transformation system. So we designed destination vectors that are, so binary vectors that are compatible with this ternary vector. And we tested a reporter gene, a reporter construct with GUS to look at transient transformation. So this is the delivery of the tDNA 7 days, let's say, after transformation. And what you see here on the right are immature embryos of maize, either with a normal typical binary vector, only the binary vector, or then a binary vector in addition to a ternary vector. And what you can see from these images is that indeed you have many more cells that are expressing the GUS transgene, but also if you look at the embryos that are expressing some GUS, this is also increased. So So that at least the transient transformation from these images is much improved when you use these ternary vector systems. Now, we also then looked at if this also translates then to increased stable transformation. So if we indeed get also more transgenic plants out of that, and indeed that is the case. And so we also see a nice increase in stable transformation. This is depicted here in this graph. And importantly, this is also very reproducible, and because if you look at these graphs, each dot actually represents an independent experiment. And so you see also that between experiments, you have a lot of variation. And some experiments, you will have a higher efficiency of transformation compared to others. And with the ternary vector, overall efficiency increases, but you still have this large variation. And this is also, I think, very clear if you look at the figure here in the middle, where we plotted the efficiency of transformation. And so here, low efficiency in yellow, high efficiency in green, over time. And each square is actually an independent experiment. And then you see that you need to have periods where you have much lower efficiency and much higher efficiency, but also within a single week, actually, you can have variation depending on where your immature embryos, from which plant your immature embryos were derived from. So in general, a lot of variation within transformation efficiency. Now, these ternary vectors help increase overall efficiency, but the quality seems to be the same. And so here on the right, we then also question, okay, you have this increased transformation efficiency, but maybe your quality goes down, and the quality of transgenics is actually determined by the tDNA copy number, and where in an ideal case, you have a single tDNA insertion of a single tDNA. So, and then if you look at copy number, comparing binary and ternary vectors, you see indeed variation in that, but overall, the distribution seems very similar. So quality is unaffected. So ternary vectors improve the delivery of transgenes using Agrobacterium. But what about regeneration? And so one of the next steps is then that you have to regenerate transgenic plants from your tissue culture. And there, of course, the last years, it's very clear that morphogenic genes, morphogenic regulators, are very helpful in that. So morphogenic regulators are transcription factors that play a role in these processes of regeneration, somatic embryogenesis, and are being used here to improve regeneration. And this work actually, the real breakthrough came actually by researchers from Corteva that showed that the combination of baby boom and mushel really improved maize transformation. And that's also something we then tried in our variety that we use, B104, and we made our own versions of their constructs that's depicted here, where you have your morphogenic genes, mushel and baby boom, controlled by very tissue-specific promoters, because you don't want baby boom and mushel to be expressed all the time in your plant later on, and that would lead to effects that you do not want, pleiotropic effects. So we tested this, if it would work, and indeed we also could show, and this was published in 2022, that indeed also in our hands, this leads to rapid formation of somatic embryos, also then leading to rapid formation of transgenic plantlets. So we could increase overall transformation efficiency using these morphogenic genes. But despite using these tissue-specific promoters, we still observe these pleiotropic effects. So plants that regenerated, they looked very unhappy. But you had a range of that, which is depicted here, from going very badly affected to almost wild type. And we could link that to copy number also here. And so when you have a lot of copies of these morphogenic genes, we saw that you had these worse pleiotropic effects. So to circumvent that, we then use this 3-LOX excision system, where we now have again our morphogenic genes, so Wuschel and baby boom, and we flank them by LOX sites. And then by using also a Cre recombinase that is inducible in tissue culture, we could then at some moment excise our morphogenic genes, leaving only our DNA of interest. So we tested that, and then indeed we still have a high efficiency of transformation, but now, as you can see, all plants are now healthy and fertile. Now, interestingly enough, many of these plants still contained the morphogenic gene, so the excision was not 100% efficient, but nevertheless, we also saw that the copy number was drastically reduced in these plants, linking it to this normal fertility. Now, of course, so this is all maize that I was explaining. What about sorghum? So we got in contact with the people at GRF in Barcelona, so the group of Ana Caño, and Juan, who is I think also in the audience, I think here today, was a PhD student in the lab of Ana, and he was interested in transformation of sorghum. And so he visited our lab last year, and he tried, okay, all what we have learned now, use of morphogenic genes, use of ternary vectors, can we now apply that to sorghum transformation as well? So we use the same setup more or less, ternary vectors that we used before. And one thing that Juan did at the start of his project was really to optimize Agrobacterium infection. And so he optimized all kinds of parameters. And one interesting thing that he observed was typically the pH is at the first steps of tissue culture is very low, because a low pH is needed to activate virulence genes of Agrobacterium. But in case of sorghum, the tissue culture did not like this low pH very much, these explants. And so he could optimize these conditions to get very nice transient expression of the GUS reporter gene. So that then also translated then, so he also used then, of course, baby Wuschel construct, which is very similar to the one we used before. Maybe one difference is the use of the kanamycin resistance marker compared to previously we were using hygromycin or imazapyr. And he could indeed get a very nice formation of somatic embryos, of regeneration of transgenic plantlets, leading to, depending how you calculate it, if you can calculate transformation frequency as the number of explants that give one or more transgenics, we get 28% of efficiency. Or if you just count all transgenics we obtain, and we get even a much higher percentage there, with one explant giving more transgenic shoots. So a very, very successful project that we also published then last year. Now, I talked today about baby boom Wuschel, which is of course the most famous morphogenic regulators, but in a couple of years ago, it was actually by accident a bit discovered by Juan de Bernardi at UC Davis, that if you express a fusion of GRF4 and GIF1 in wheat, you get much higher regeneration frequency and much higher transformation frequency compared to control. So this is actually an alternative morphogenic genes that are used in wheat, and they approached us to see that it would also work in maize. And this is actually work of the PhD student Wout van der Putte at VRB, who tested then, okay, can we use GRF-GIF in maize? And we tested then both the wheat version, but also a maize version, so using GRF-GIF of maize. And indeed, we could also see that in maize, this leads to a higher transformation efficiency. Now, the difference with Baby Wushel, which is very interesting, is that you do not have these pleiotropic effects. So even when you have expression of GRF-GIF, you do not see any negative effects on plant development, on fertility. And this is now something that Wout is testing also in different inbred lines, if he can use that also to use it in those genotypes. So one thing we did already in B104 is we try to combine it with gene editing. So here we are targeting a gene called chaos that is involved in chloroplasts development. And if you knock out this gene, you get these pale yellow leaf phenotypes. And so we could see that CRISPR combined with GRF-GIF works as normal and very efficient, while keeping still this increase in transformation efficiency. And so very nice results there. Now, of course, again, this is maize. Does this translate into sorghum? So this is not something that we have done, but a bit at the same time that Wout was working on this and the use of GRF-GIF in maize, there was this publication where they tested this in sorghum. And it's a very similar setup as we have tried, so also the same Ternary vector, but then indeed using GRF-GIF as the morpho gene in sorghum, and it's a bit of a different version with a wheat GRF4 and a rice GIF1. And so also in sorghum, this indeed then led to a higher transformation efficiency. So also very promising there. So what about gene editing? So in the work of Juan, we didn't yet test gene editing, but if you look in the literature in the use of gene editing in sorghum, there are some very interesting recent publications there. So there's the group of Peggy Lemeau in the US, who has been very active also in this field, also using the combination of Baby Wushel in sorghum before in collaboration with Corteva. And recently they have this preprint out at the moment where they are using a new version where they're using only Wushel and that seems to be efficient enough to improve sorghum transformation. And then on the gene editing side, they show that they get very high efficiency editing using this version of Cas9, which is intronized. So this is a Cas9 coding sequence with the addition of one or more introns. And this is something that we see more and more now in the field, that people are using these intronized Cas9 variants. All right. So maybe a final subject of today is the use of these explants. And so all the work that I showed until now are all starting from immature embryos as the explants. And these are quite tedious to obtain. And so in the case of maize, this requires the continuous growing of wild type plants that you have to pollinate, that you have to, at a certain time of the pollination, you have then to isolate the embryos from the cobs. And the quality of these embryos and the quality of the plants they are derived from is very essential. And so you need a very nice infrastructure to obtain such immature embryos. And it's the same holds true actually for sorghum and even for wheat. And obtaining high quality starting material is very important. And this has been a major bottleneck. Okay, so just historical at VIB, people used 600 embryos for transformation of one construct. Now, with all these improvements that we could make over the years, this has dropped to 150 per construct, more or less, but still this is of course quite laborious and creates variation. So can there be an alternative? And also here, people from Corteva who worked on Baby Wushel also test, okay, can we use then these morphogenic genes also to transform and regenerate from other explants. And leaf fragments are a very interesting system there. And so the idea is depicted here. And so where you of course start just from seeds, you germinate seedlings in vitro, and then you take the base of the seedling and you literally blend them into these small fragments that you then also co-cultivate with Agrobacterium. And again, this is a combination of having a very good Agrobacterium-mediated transformation system using ternary vectors, and then these morphogenic genes like baby boom, which then lead to somatic embryogenesis on your leaf fragments, leading then to transgenic seedlings. Now, interestingly, is in this paper from Corteva, they also include sorghum there, and also show that also there, starting from leaf fragments, they could obtain transgenic plants. And so, especially I think for labs that do not have these extensive greenhouse facilities, I think having such a system would be very, very helpful. So we ourselves, so we also were interested in this. And so we have a collaboration with GCCRC, which is a joint venture between Embrapa and the University of Campinas in Brazil. And Leticia Rios, who's I think also joining today, was interested in testing leaf transformation for their tropical maize varieties. And so working with different varieties poses additional problems because these tropical varieties might be difficult to pollinate in your greenhouse, really controlling the formation of high quality immature embryos. So starting from leaf explants there was very interesting. So again, we used the knowledge that we had, ternary vector, morphogenic genes, a construct which is very similar, small differences with the constructs we used before, and tried if we could also transform leaf fragments. This was also, to cut a very long story short, Leticia also optimized many steps in this regard, but could then show, and these are each row here in this image is a different tropical maize line. We could show indeed transient expression of the Gus reporter gene, also the formation of somatic embryos and transgenic somatic embryos that also could lead to transgenic tropical maize plants. So very promising. Still, let's say, a lot of variation between genotypes, very clearly. And overall, I think efficiency can be improved, but very promising results also. Now, what about sorghum then? So I showed you that people of Corteva also showed that it could work with sorghum. But very recently, again, there was a preprint from the Voitas lab that also showed that they could use leaf transformation using sorghum. So you see here their picture, which is again very similar to the one of Corteva and ours, where they use these seedlings for blending. They could show it would work with baby boom Wushro delivered as binding vectors, but they could also combine it with viral vectors. And the Voitas lab has this interest in viral delivery of transgenes. And what they did is they had their pDNA with a gene of interest, here just a blue fluorescent protein, and then a vector to aid for transformation using the morphogenic genes incorporated in a viral construct. And they could then show that they had indeed a higher transformation efficiency and also the generation of transgenic plants that are viral, free of virus. And so this is something to check out if you're interested in that. So maybe to summarize, and this is actually a figure I did not make myself. So there's actually a very recently a review paper on sorghum transformation that I advise you to check out. And also another interesting review on this subject is from the Canwan group, which is also has been very active in this field, especially in maize. To summarize, typically for sorghum, we use immature embryos as the explant. Conventionally, this takes quite a long time with low efficiency or no efficiency at all. But then, of course, using morphogenic genes, this can be improved and also ternary vectors for delivery. Now, and then, of course, upcoming is the use of these alternative explants, leaf explants that are more readily available, and also this combination then with morphogenic genes. So then, of course, all of this work was done over the last years at the crop genome engineering facility at PSB, at VIB. The crop genome engineering facility is now headed by Lennart Hoegenaert, and they are really specialized in maize and soybean transformation, and also offering that as a service. So both to academic groups or to companies, really working with many labs all over Europe, but also over the world. So if you're interested in that, please check out their website. It's actually cgf.be, and the E is here. So check that out. So I myself, as Mark introduced, I moved last year to the Faculty of Bioscience Engineering at Ghent University. I joined my colleagues here at the Department of Biotechnology, at the Molecular Biotechnology Cluster. And the research in my group is still focused on all the aspects that I just mentioned, and on improving agrobacterium-mediated delivery, plant transformation, and gene editing. Of course, some words of thanks, of course, to the entire Center for Plant Systems Biology, but in particularly, I think Wout van der Putte, whose work on GRF-GIF and ternary vectors I presented, Griet and Stan, who are really the experts in maize transformation in that lab, and also Jari, who was a technician in the lab, and during that period also helped a lot with these experiments. Then our collaborators, the lab of Anya Canio in Barcelona, and especially Juan, who did the work on sorghum transformation, Juan de Bernardi at UC Davis, and then our collaborators at Campinas, and especially Leticia. And then you see here on the bottom, you see this agrobacterium here, and that's to remind myself that there's another slide. I want to also make a little bit of advertisement for the Agrobacterium meeting. So every year there is the Crown Gall meeting in the US, but this year it's coming again to Europe. And so together with a number of labs in Belgium, we are organizing the Agrobacterium 2025 meeting in Leuven in Belgium. And so I'm very happy. So this is the first time I can announce this. We have a first keynote speaker that can be confirmed, which is Patrick Shih from UC Berkeley, who is doing really exciting work on combining synthetic biology and agrobacterium, and he will be speaking. And I'm so really looking forward to that talk. And so if you're interested, abstract deadline is May 1st, and you can find more information there. So that's it. So I end here, and if there are any questions I see in the chat, there are already some, I'm happy to answer them.
Thank you, Laurens, for this very nice presentation. Indeed, there are no questions yet in the chat, but please do post them there if you want. You can also raise your hand and ask the question directly, I think. I don't know if there are any for the moment, but maybe I can start because people are a bit shy to ask questions, Laurens. I have a few, but if I understand well, you have been working mainly on maize, and we also see this thing on sorghum. But if I heard it well, it's not always the same efficiency also among maize accessions. You have big trouble in transforming some maize accessions.
Yes.
Is there any explanation for that? Because I expect you have now some effectors and so on that are helping you. How does it come that you have this discrepancy within the same species, let's say?
Yeah. So clearly, yeah. So all the work I except of course of the work of Leticia is in the B104 variety, which is actually chosen to be a variety which is easily transformable. And so you will see that typically also within industry, people have been working in certain varieties internally that they know that are easily transformable. And there are two aspects on that. You have the susceptibility to Agrobacterium, which is the first aspect, and then you have the regeneration capacity. And both of them are very dependent on the genotype. So it can be that you have a variety which you can indeed have transient transformation, so it's susceptible to Agrobacterium, but then later on it will not regenerate very well, or vice versa. So you have to have both of them correct, and this is of course very challenging. And the methods that we are now using, these can help that, but will not eliminate this completely. And so we are not yet at the stage where we have genotype independent transformation, not at all. So I think the term that has been launched by the Kanban group, I think, is genotype flexibility. So you have it's not complete independence, but you can at least have more genotypes you can transform. But this is still a major bottleneck, even with all the tools we have now at hand.
Okay, thank you. I see Steve, you have a question, please.
Yeah.
You're muted.
Yeah, thank you, Lawrence. I just have a quick question. I don't know if I missed it. How do you excise, or what activates the excision of baby boom bushel?
Yeah, I went over that a little bit quickly. I know. So there are a couple of ways in which it is done. So in the system, the initial system that we used when we use immature embryos as explants, it's actually a promoter that is thought to be induced, that will automatically be activated later in embryogenesis. It's a GLB promoter, but also is activated by ABA. And at some stages in tissue culture, we will add ABA in the tissue culture media, and that will also induce this promoter and hence the Cre recombinase. In the case of leaf transformation, where we are also using this excision system, it's a heat shock promoter. So it's by putting the tissue culture in at a couple of hours at a higher temperature that is used. The thing is that I think improvement here is still possible. I think the heat shock we know is very it's also very variable, let's say, also not always easy to reproduce that. So I think if we would have a better inducible system, that would really help. But unfortunately, in general, in plants, having good inducible systems is not so trivial. So that's indeed a point of improvement, I think, still. Yeah.
Thanks. And one more question. I saw the explants for leaf transformation were quite short. Are they treated in any way to make them dwarf? Because in the protocol that I read on the Nature paper, they inhibit with GA.
Yeah.
So is that the same?
We're using the same, yeah. So it's ansimidol we are using, and that's indeed a compound that inhibits GA biosynthesis. Yeah.
Thanks.
Okay. Lawrence, we do have some questions in the chat. I will read them for you, maybe from Nuan. That person is asking if you can comment on the comparison of number of somatic embryos and shoots by both transformation methods, meaning embryo and leaf phases.
Yeah. The fact is that I think at least in our hands, the leaf transformation is still, and I think in general, leaf transformation is still a method which is being developed. So I think at the moment, this is much less efficient than transformation using immature embryos. So this needs to be improved. I cannot really come up with exact numbers there, but it's really I would say a factor of 10 less compared to immature embryos. I know that some groups have really good success with that, but I think also there you have to see how reproducible all of that is, and that's a very important aspect of this.
Yeah, please.
Yeah, because in the question there was also this about the number of somatic embryos. It's also an interesting I don't know if that was meant with the question, but in general, in both methods, you see actually a lot of somatic embryos forming, but also only very few of them will lead to transgenic plants. This is still a little bit of a mystery in the field, and the idea is that maybe combining morphogenic genes like baby boom and GRF-GIF, that this would help with that you have indeed baby boom will increase the number of somatic embryos, but GRF-GIF will maybe act later in the development and maybe lead to more of them really germinating and leading to transgenic crops. So a couple of groups are also working in combining morphogenic genes together, and so we are seeing the first results of that also appearing in the literature.
And following up on these questions, we have a question from Mona, and she's asking about pollen transformation.
Pollen transformation, yeah. Okay, yeah. So I think she refers to a paper that was published a couple of years ago, where they indeed showed that you could combine pollen of plants or a variety of plant species with DNA that is coated on nanoparticles, and then the DNA will enter the pollen, and if you use that for pollination of plants, a low percentage, but still would be transgenic. This, of course, would avoid any tissue culture and would avoid all of these problems that we have. It does not involve agrobacterium, it does not involve tissue culture. Unfortunately, this has not been reproducible. And so a lot of labs have tried, and there is even a paper, it was originally a preprint, but I think it's now published, of a number of labs that report actually their failure to reproduce that work. And so at the moment, pollen transformation is not yet a reality, but maybe in the future people will indeed be able to make it so. And so that would indeed be a very important aspect. I hope that she uses, she means that with pollen transformation, but because you also have other systems that may be related to that.
Okay, she can still complete on that question for the final question and answer session. And now I propose that we move on then for the next talk that we will have with Stephen Runo. Stephen Runo is Associate Professor of Molecular Biology at Kenyatta University in Nairobi, specializing in plant molecular biology and genetic resistance to parasitic weeds. He got his PhD in plant molecular biology from Kenyatta University and the University of California, Davis, on resistance mechanism against parasitic weeds. His research focuses on developing Striga-resistant sorghum using molecular genetics, genome editing, and breeding technologies. Through his work, he has successfully advanced Striga smart sorghum, now at an advanced stage of development, leveraging resistance breeding, molecular assisted breeding, and genome editing. The 2020 Royal Society of Africa Prize recognized his outstanding contributions to African agriculture through science. And as a leader in international collaborations, his work bridges scientific research and agricultural application, ensuring real impact on food security and farmer resilience in Africa. With this, Stephen, if you want to share your slides, we are happy to hear from you. And you are still muted for now.
Thank you, Mark, and thank you, Lawrence. You did a lot of groundwork for me. So I'm just going to talk about the applications of agrobacterium-mediated transformation, because that is what we use for practical applications of genetics in developing resistance against parasitic plants, and the parasitic plant that we work with is Striga. So in my talk, I'm just going to give a very brief overview of what Striga is and why it is difficult to control Striga using conventional methods. And then I'll talk about the mechanisms of Striga resistance, because we need to identify mechanisms of Striga resistance. and the genetic underpinnings of these resistances so that you can be able to use molecular genetics to advance or to impart resistance against Striga. Not only to impart resistance, but also to understand the genetics of interactions between Striga and its host. And we do this in mostly all grass cereals, rice, maize, and sorghum, as well as finger millet and pearl millet. But today I will focus the discussion on just what we are doing in rice, what we have done in maize, and what we are doing in sorghum. And then I'll very lightly touch on what the regulatory landscape is in Kenya for gene-edited products, because eventually all our research is destined to have a translational impact, and we try to find the shortest route to commercialization and deployment. Then I'll just give conclusions at the end. So for those who don't understand Striga is this plant that I'm showing on my screen. It is the purple colored plant, and this farmer planted maize actually in this farm. So this is a farm run and overtaken by Striga infestation. We estimate that Striga causes about 30 to 100% losses. But as you can see from this photograph, this is not even 30%. This is most likely 90 to 100% in yield losses. And we estimate that about 400,000 hectares of land are infested in Africa, sub-Saharan Africa, and this causes about $7 billion in losses annually. I think I want to emphasize here that these are all statistics. The crop losses due to Striga are much, much higher now, because we know that Striga has expanded in its natural range. So there's a lot more land that has been infested. Resistance that has been deployed is much, much lower. And so we have a lot more losses, more than $7 billion. But for now, that's the statistic that we have. And there are 3 specific species of Striga that are more dangerous and more of economic importance: Striga montica and Striga asiatica, as well as Striga gesneroides. The cereals, so maize, millet, and sorghum, are afflicted by Striga montica and Striga asiatica. Striga gesneroides is a pathogen of dicot and especially cowpea. And I just want to map this on the Africa distribution of crops that are grown in Africa, the staple crops, maize, millet, and sorghum, and then map that with the distribution of Striga. So as you can see, Striga is prevalent in all parts of sub-Saharan Africa. And if you don't have one species of Striga, then you have the other one. And it's very unlikely that you'd have low Striga infestation in any part of sub-Saharan Africa. And like I said, this range is expanding very, very rapidly. And with the climate change, you expect this to expand in most other places. One of the reasons that Striga is a very successful parasite is because Striga has been able to synchronize its life cycle with the host. So Striga only germinate in the presence of germination stimulants sent by the host. And these seeds are greatly enhanced. This is 200 micrometers if you look at this video. So the scale shows that Striga seeds are really, really small. And this can stay in soil for decades. If you're familiar with the Arabidopsis seeds, this is maybe 10 times or more than 10 times smaller than Arabidopsis seeds. And they rely on germination stimulants that are released by Striga. And upon perception of those germination stimulants, Striga is able to very, very rapidly germinate. A period of less than 6 to 8 hours, Striga has already perceived the germination stimulant. And after perception of the germination stimulant, Striga is able to infect a host led by chemotaxis. It is able to locate a host root, and then it grows inside the host roots and continues growing until it establishes a vascular connection. At the point of vascular connection, what we know now is that Striga would be delivering phytotoxins as well as effectors that help it colonize the plant. Acute, this process also takes a very short time, less than 36 to 48 hours, the infection cycle is complete, and there are vascular connections between Striga and its host. And at this point, it's really difficult to manage the host or develop interventions on the host because it undergoes reprogramming and there's growth retardation and yields are compromised, in fact, at this point. So what we do in our lab is to try and understand mechanisms of these interactions. We rely a lot on natural sources of resistance, and I'll start with rice. We know for a long time it has been known that rice has some levels of resistance, and one of the rice varieties that we know to be resistant to Striga is Nipombare. We have a soil-free system that we use to assay for resistance responses, which I'm showing in the slide here. So this is a highly susceptible rice variety. You see that it has attachments. These attachments here are Striga attaching on the roots of rice. But on the same level of infestation, you see here that there's no attachments here on this variety of rice. And so we know that there are resistance mechanisms, and we can be able to quantify that using Striga attachments, Striga length, and Striga biomass. You can be able to get an estimation of how much Striga has attached on a crop or on a variety. And then from there, we try and decipher what these mechanisms is. Our choice method of choice is using omic technology. So we do genome-wide association studies, but we also use transcriptome analysis to try and understand what pathways are activated. So I'm showing an example of such study where we did a transcriptome analysis between rice and a susceptible rice and a resistant rice. And what we were able to figure out was general mechanisms of resistance. So most of these are cell wall based, so addition or lignification or cellulose biosynthesis and typical plant pathogen interactions. But the bulk of the resistance in Striga against in rice, Nipombare rice, was actually a cell wall mediated resistance. And here is an example of that. And this is lignification assayed here using the test for fluoroglucinol staining. So you have a highly susceptible variety here where Striga infects and forms a very clear attachment here. This is the xylem of the host, and then this is the parasite. And then this is a transverse section showing Striga going all the way and attaching to the host. And then this is a totally in blue version of that. And you can see that there's very clear host-parasite interactions. But in the susceptible variety, you have Striga not attaching very well. And in fact, you can see that Striga tries to go in and then exits on this other side. And a more clearer version here of the totally in blue is that unlike in the susceptible variety, you don't have interactions between host and parasite. And in most cases, the parasite will die. And that explains most of the resistance in rice. And this was sort of expected because this has been documented in rice. But a closer look at the transcriptome showed that it is not necessarily the genes that were involved in disease response, not typical host pathogen interaction genes, although those were there, and not typical cell wall response genes, although those were also enriched. What came to surprise us was that some of the genes that were highly induced were genes that were involved in phytoalexin biosynthesis. Phytoalexin, that would mean that the genes that would be producing pathways that are activated to produce secondary metabolites that would be acting against Striga infestation. So this has not been documented in rice, even though we know that phytoalexins are important in causing disease resistance or responses against crop pathogens. And then a closer look at the genes that were induced and a little literature search showed that most of these genes are clustered in a chromosomal region in rice. So these were inherited a long time ago or were transmitted into rice from long ago progenitors, and these are arranged in a cluster. So what we did was to look at all those genes again and then profile them between Nipombare and the susceptible variety. And what we were able to identify is that in Nipombare, all the time, these genes were greatly induced compared to the susceptible variety. And that made us to form the allelopathy hypothesis as a resistance mechanism. So one of the students that now is not in the lab anymore, but did these experiments on germination assays of Striga using root exudate from Nipombare, the susceptible variety, the resistant variety from the susceptible variety, and from GR24, which is an inducer of Striga germination. So in Nipombare, in all times, or in most cases, you don't have induction of germination, but in the susceptible variety, you have high induction of germination. Okay, so that's fine. It is possible, but that maybe Nipombare does not have high germination stimulants, but it is also possible that Nipombare is inhibiting germination through products of the root exudate. And so to separate out these two hypotheses, we again used Nipombare root exudate and spiked Nipombare root exudate on germination on root exudate of IAC and GR24. And so we saw a reduction in germination and attributed this, in fact, to the allelopathy. So we go back to the allelopathy and the pathways that lead to production of these molecules, phytoalexins. And in fact, one of the major allelopathic chemical in sorghum, and in particularly in Nipombare, momilactons. And as you can see here, one of the genes that was induced was one of the genes that was at the critical stages of momilacton biosynthesis. And so this led us to form the hypothesis that if you're able to flux or direct biosynthesis of momilactons, as in Nipombare, then you are likely to have a higher resistance. And one of the critical genes is this gene here, and then CPS1, which leads to other phytoalexins. And so what we wanted to do was to divert that biosynthesis to this flux. And that would mean you knock out one of these genes. So Maki, one of the genes is involved in GA biosynthesis, so you don't want to mess with that. So you need to pick the one that does not involve GA biosynthesis. So we partnered with Peter Rubin of ISU, Iowa State University, and Rubin provided us with mutants or CRISPR mutants of CPS1 and CPS1 and also this gene here that was CPS4, just to confirm that change in flux in momilactons would change Striga biosynthesis and also momilacton biosynthesis and also impart Striga resistance. So this is work that is still on progress, but we hope to decipher this more. And if this is indeed the case, then this can be a way of using, changing the flux of metabolites to achieve Striga resistance in rice. And then we have work that is ongoing on in maize. In maize, again, we try to identify sources of resistance. Some of this has not been documented. We don't have good data to document genetic causes of resistance in maize. But a few years ago, 1 year ago, there's a paper that came out from Hoaro Bomister's group that showed that maize exhibits differences in variation of in germination induction, and this was attributed to some genes that were involved in the strigolactone biosynthesis. This is work of one of my PhD students that just started, and what she did was to evaluate maize varieties and then collect root exudate from them and then induce germination. What we want to do is to profile the different germination stimulant efficiencies of maize. And just to draw your attention here, this is one of the varieties that is highly resistant. EH14 is highly resistant to Striga, meaning that it produces very low germination stimulation effect compared to these others. What we also did was to go in on the paper that was published by Harrow's group and identify the genes that were responsible for biosynthesis of strigolactone. And one of the genes is MAX1, and then the other gene is CLAMP1. So variations or mutations in these genes are expected as was reported in that paper, to cause differences in strigolactone biosynthesis and hence strigolactone germination stimulation and hence Striga resistance. So from the batch of genotypes that were collected and determined for germination stimulation, we were able to determine that some were highly resistant than others. And then what we were able to also do was to quantify the levels of strigolactones between the different varieties of maize. Long story short, the ones that were highly resistant were producing one type of strigolactone compared to the ones that were susceptible producing another kind of strigolactone. And this is work that was done in conjunction with a lab in King Abdallah University of Science and Technology. And here is the summary of the work. If you quantify zeolactone, one of the strigolactones, and if you quantify zeolactone, one of the other strigolactones, then the resistant varieties would always have high levels of zeolactone compared to the susceptible varieties. So you have EH14 producing very low zeolactone, and then the ones that are highly resistant, highly susceptible, producing very high levels of zeolactone. And so we went back to the hypothesis that it's possible to change flux again of zeolactone production versus zeolactone production. And just get here that the more zeolactone you have, the more susceptible, and the more zeolactone you have, the less susceptible. And if you knock out those two genes, then you're able to change flux and have more zeolactone produced instead of zeolactone getting produced. But the bottleneck to that, and one of the things that we wanted to do, is to take advantage of the rapidly advancing methods of genetic transformation. And so what we did was to partner with Lina Tripathi, who's already well versed with methods of genetic transformation, and Grace, who's the student working with me and Lina, used the protocol that was developed by Koteva, the Wang et al. Leaf transformation protocol, that was also very well explained by Lawrence. So step by step, and was able to reproduce, or was able to have high efficiency or decent enough efficiency of transformation of leaf explants. This is just a test construct. There is no guide here, but what we hope to do is to use guide RNAs to impart resistance using some of the genes that we identified, or one of the genes that we identified as being important in strigolactone, as well as Striga resistance in maize. And hopefully we'll be able to produce edits quite soon. Then lastly, the sorghum work. Unlike maize, unlike rice, Striga resistance in sorghum is very well characterized. And this work started a long time ago in 2000 to 2009 by Jeta Gebisa, who was working on Striga resistance in sorghum. Jeta went on to win the World Food Prize because of his work in identifying and breeding new varieties of sorghum. At that time, it was just known that some sorghum varieties were resistant to Striga. So as you can see here, there's no Striga infestation, but there's a lot of Striga infestation here. So get that the field is homogeneously infested by Striga, but some varieties would have more resistance than others. And so fast track to 2017, the same lab, Ijeta's lab, was able to map the resistance of this variety of sorghum called SRN39 to a chromosomal region in sorghum, chromosome 5. And it was established that some varieties of sorghum, this variety of sorghum that he was working on, had a deletion on that region. And this region came to be known as the low germination loci. And then much further ahead, it was identified that the causative gene for this resistance was a sulfotransferase that played in the interconversion of one form of strigolactone into another form of strigolactone. So this is not zeolactone. This is not zeolactol. This is a strigolactone specific for sorghum, 5-deoxystrigol, and orobancol. And so my PhD student at the time, Sylvia, went on and identified primers that we can use to map these genetic regions and see if you can identify more genetic mutations or mutants of sorghum that would have this deletion. Sort of about 300 and so genotypes of sorghum. We were able to identify about a dozen that had a mutation. And the corresponding mutation had, we measured the stregolactons, and they were consistent with the chemotype being, if you have a deletion, then you have low germination stimulation. And if you don't have a mutation, you have high germination stimulation. We were able to take this to the field. This is not one of the pictures for the LGS1, but we were able to show that the more the highly resistant genotypes that did not have LGS1 were also consistent with low Striga infestations. And then this brought us the idea of using CRISPR to recreate mutations that were identified by Ijeta. Again, a very straightforward experiment. The hypothesis here is that if you remove the region that causes Striga germination, then you could recreate mutations that would have low germination stimulation using CRISPR. So we partnered with Corteva to do this work, and we were able to identify and produce CRISPR mutants that were Striga resistant. We developed, or these mutations were developed quite rapidly and were also advanced quite rapidly because of the way that they were developed. So on my next slide, I'll talk about germination. I'll talk about CRISPR regulations or regulation of gene-edited products. But before I get there, the edits were developed in such a way that they did not have any remnants of the CRISPR machinery. So they were DNA free of any genetic elements aside from sorghum. And so with the biosafety laws in Kenya, we applied to have them tested in the field as if they were non-GM. And so we were able to advance very, very quickly from target identification to development of edits to field evaluation. So this is just one of the, this is a letter that we got from the NBA to tell us that you can, in fact, do genome-edited products because we were able to prove that there were no remnants of the Cas9. So we were able to rapidly advance to the field. And here is one of the, or an example of the results that we did last year. So here you have the edited version, and then here you have the non-edited version. And we're able to see a reduction in Striga infestation and also a restoration in yield or an increase in yield because of less Striga infestation. So this is encouraging, and this tells us that we can be able to develop CRISPR edits quite rapidly and then advance them, do POC or proof of concept in the lab, and then rapidly advance those to the field. And then if they are good and stellar products, then the route to commercialization is quite rapid compared to other technologies or the conventional GM technology. So what we do, in addition to field testing, is to try and actively look for really good targets that we can impart using CRISPR. We like CRISPR because it is in our best interest to advance this as fast as possible, and because it has less regulatory hurdles. And then the other thing we do is to try and optimize and make sure that we have really good, neat working protocols for validation, for genetics, to test the genetics, and also to develop products for deployment. Our future goal is to have not just one level of resistance. I think for us, we want to have at least 2 or 3 levels of resistance. We have some targets that we have started to identify that we want to stack and have the genes deployed in tandem 2 or at least 3 levels of resistance. So that's what we're still working on. So this is the slide that I'm talking about. And I need to update this because there's a seventh country. We have 6 countries in this map: Burkina Faso, Ethiopia, Ghana, Kenya, Malawi, and Nigeria that have gene editing specific guidelines. And these guidelines are very similar in the way that they have been developed, meaning that it's possible to test one variety that has been cleared in one country and developed and then tested in another country. So Zambia just joined, so we have the 7th country, and expect that more countries will be will have gene-edited guidelines. And as much as possible, what we also like to do is look for varieties of sorghum that are popular across these countries, and those that have Striga infestation, so that we develop the same varieties that would be popular for scalability, because it would be important to scale this or to upscale this, and also to have an initial investment in terms of investment in technology, but it's easier to develop and upscale that technology to more than one country. We also try to develop this into varieties of sorghum that have commercial value, so that there is good pull-through demand for them. So I think I just want to end with the conclusions here. I think we have a lot of potential now more than before. It's really hard to imagine how we have advanced really rapidly from tools of genomics to identify targets, and then to validate those targets, and then to develop near products that would be ready for deployment, or that can be ready for deployment in theory. But I also want to caution that, as I have explained, it's not a one-size-fits-all scenario. What would work for sorghum would not work for maize, would not work for rice. So there are different mechanisms. And so that's why it's really important to look out for genes and genetic reasons for continually look for genetic regions that underpin resistance. And I think omic technology, whether transcriptomics, whether genomics, whether GWAS, or whether proteomics, I think these are really powerful tools that we can combine and identify regulatory regions. And to me, this would be the rate limiting step. If we identify really good targets and we have neat methods for transformation, then I think in the not so distant future, it's possible to see in market and at beyond POC level Striga-resistant varieties. Yeah, and I think genome editing allows a more permissive, more acceptable way to develop this resistance. So I think for us, and especially for us in Kenya and in Africa, it's within our best interest to use genome editing technologies, because one, more permissive, less regulatory hurdles, and more efficient. I did not also mention that some of these methods that we are using were, in fact, difficult. It was difficult to achieve mutational breeding for sorghum with earlier technologies. So I think CRISPR and genome editing provides more flexibility and more options. I think this is my last slide on acknowledgement. There is some work that overlapped with a grant that we got from National Research Foundation in Kenya. Some of this work also overlapped with International Center for Genetic Engineering and Biotechnology. I have really good partners in IITA, as well as Penn State and Corteva, who have made the process of genetic transformation and gene editing really possible, and hopefully can be able to make these processes routine. And then a lot of thanks to PhD and master students. So Sylvia did the sorghum work, Damaris did the rice work, and the work for maize is being carried out by Grace together with Esther and Jay Tripathi and Lina Tripathi at IIT. So with that, I end, and I hand over to you, Mark.
Thank you. Thank you for your talk. I wonder if we have already some questions. There is nothing yet in the chat. Please, people, do not hesitate to write any question in the chat or yet raise your hand if you want to ask a question to both Steve to start with and also back to Lawrence if you still have questions for him. Again, it seems that people are a bit shy. I'm sure I can start with something, Steven. I was wondering if you, with these mutations, especially in the strigolactone pathway, did you face any other phenotypes, any unwanted phenotypes while mutating this? I'm thinking of, I mean, if you have resistance against Striga, but then you increase the sensitivity to others, would that happen, for example, or any other phenotypic effect that you would have with those mutations that you induce?
So the most advanced one is sorghum. Bear in mind that these are already existing mutations in nature. So they already are mutations of low germination stimulation in nature. So we already have the varieties that were identified much earlier. We don't have any adverse phenotype as we observe them in the field. So we have not seen any adverse effects. So in terms of susceptibility, one of the other things that we are checking is if there is any adverse effects in terms of colonization of roots by mycorrhiza fungi, because that's one of the chemotypes of strigolactone. So, but we don't expect there to be any. And I think there was a study that was done much earlier by the Jetta group that did not see any changes in the efficiencies of colonization by mycorrhiza fungi. I don't know about rice because, and that is something that we should also check, because phytoalexins, some of those phytoalexins may be involved in fighting for against the Magnapothe oryzae or other pathogens of pests. We don't know that yet. I think for us, firstly, is to identify and to validate the allelopathy hypothesis, and then see how we can tweak the flax for optimal either Striga resistance or for other crop pathogens. For maize, I don't think it has been checked, but if it's strigolactones for maize and sorghum, it doesn't seem to be finicky for mycorrhizal associations. There is a whole lot of strigolactones, and it doesn't seem like mycorrhiza care so much about the different isomers or the different steroid isomers of strigolactones. But it's something that needs to be checked.
Mm-hmm. You were also talking about looking for, Lawrence, you have a question, please.
Yeah, so Stephen, very nice talk, very, very interesting. And you say one size does not fit all. So, but maybe I missed it. But so LGS1 that you discovered in sorghum, this is not something you can translate to rice, then, for example. You don't have a similar gene in rice, or
No, yeah. So that's always the temptation. But unfortunately, the strigolactone biosynthesis, even though quite similar, upstream processes are quite similar, but there's a variation in the way that the resistance is exhibited in rice than in sorghum. But having said that, the basic genes, or one of the genes that is involved in strigolactone biosynthesis, one of the genes that is involved in the interconversion of zeolactone, for example, to zeolactone, an ortholog of that has been used in rice and in pearl millet, but it's not the LGS1 region. The LGS1 region is very specific to sorghum. But also, having said that, I don't know if people have seen, there's two papers have come out, one in Cell and one in Innovation, the last 3 weeks, I guess. And these papers were on strigolactone transporters. So one of the genes that we identified in a GWAS study but we did not follow up that time, was an ABCG transporter that transports strigolactones to the rhizosphere. And it just seems to be very specific for, as the two papers explain, one in rice and the other, no, in sorghum, and then the other one in tomato. It just seems very specific to be directing strigolactones to the root and not strigolactones to the above ground parts of the plant. And orthologs or homologs of those genes in sorghum, in maize, in tomato can be transferable. And so there you can find a one size fits all. So just get the ortholog of those, and then you shut down strigolactone transportation. What we don't know about that is if you completely shut down strigolactone transportation, if you're also compromising the mycorrhizal and below ground communication, which is also a really important thing. So the danger to that is to completely shut down the strigolactone exudation in the rhizosphere.
Okay, but it might then be important to get to your 3 levels of resistance, right? To have these
yeah. So apparently it's a really good it's from what I read, it's a very powerful gene. And if you have a double knockout, because there are 2 transporters, ABCG36 and ABCG48 orthologs, then you limit strigolactone exudation. One of the other things you can do is to have amendments, is to have natural or artificial addition of P and N in the soil, and complement that with the residue.
I was wondering, Steve, so you, like, for example, for sorghum, you then managed to have quite a good resistance to Striga. Is that also very specific? Would you have another parasitic wheat in the field, let's say, would that then still be an issue, or is it kind of a more general mechanism that allows resistance to several parasitic weeds?
Yeah, it's rare that you'd have an overlap of 2 parasitic weeds. It's quite rare that you'd have 2 of them together. But the mechanism would be the same, less germination stimulation. All parasitic weeds are stimulated with Striga harmonica and Striga asiatica. They're both stimulated by the same strigolactones. So it would work, but you rarely have the same infestation. But also, I think I now get, maybe this is what you mean. If you have Striga asiatica in Kenya and you have Striga harmonica in Kenya, would they work the same way? Yes.
Yeah, the main question was like, if you reduce the possibility of one to be parasitic, does this, let's say, allow the emergence of the other? But as you say, no, probably no, right?
No.
Okay. I still had a question actually for Lawrence also. So you have been showing these different effectors, so baby boom and bushel, and then this GRF-GIF1 or something like this. Is there a way, or is it even useful to try to find other effectors, and how would we screen for that?
Yeah, I think it's a very good question. Huh? So it is indeed of interest also to my research at the moment, to indeed come up with additional morphogenic genes that can be used maybe in combination, or maybe to come back to the question you had earlier, if the current morphogenic genes might not work for a certain genotype, but maybe you will find another morphogenic regulator that works for that particular genotype. So I think there is still a lot of room to discover new ones, and we see also in the literature that people are finding indeed new morphogenic regulators, and I think they can come from 2 different angles. I think like Stephen has shown to use omics, but then in this area of regeneration to build networks and build hypotheses like that. And the other way they come from is really more indeed also a bit what was discussed there, like GWAS studies, but then on regeneration. So there's this very famous work that has been done in maize, where for many years, people have looked at two different maize varieties, one regenerating very easily, the other one more difficult, and then to come up indeed with the causative, the gene that is causing this difference. And it ended up now recently to be a wuschel-like protein, not wuschel that we have been using, but a family member that was actually the cause of this difference. So this is then an alternative. It is related, it's in the same family, but it's related. But I think indeed also from omics data, I think it would be feasible, I think, to come up with other factors. I'm very convinced that it's not limited to baby boom and wuschel here or GRF. I think you will see in the future that more and more alternative ways, transcription factors likely, but maybe also non-transcription factors will be identified that can help with that. Yeah.
But we would expect that to be quite similar in different types of crops, right? I mean, you would still have probably the same effectors working in several crops, or
Yeah.
you think there would be a difference?
I think we have to make a distinction here, especially between monocots and dicots. So monocots, typically regeneration works through somatic embryogenesis. So there indeed baby boom and baby boom-like proteins have a major role. For dicots, typically there the regeneration protocols that we use in transformation are actually based on organogenesis. And there it might be that the factors we will identify are quite different. So we will have to see how that turns out, I think. Yeah, but I think I can imagine that these might be quite different there. So. Although I must say GRF-GIF is actually an example, I think, of a factor that has been shown to work in both very clearly. And so there was a recent publication on tomato actually, where they have used GRF-GIF also to improve transformation.
So this one seems more ubiquitous, let's say, than others maybe. Yeah. Let me go back a bit to the field with questions for Steve. I see in the chat we have a question from Jacinta. She's asking, what other traits are you interested in that you want to stuck them together with the Striga, basically combining of stress resistance? What would be like your favorite combination of disease or stress resistance for the specific case of sorghum, I suppose, in Kenya?
Yeah, so what I did not say is that there's a precedence to this. We had the same way we had a transcriptome in rice, we had a transcriptome in sorghum, not just 2 sorghum varieties, but this was a much bigger study that had 5 varieties of sorghum that have different mechanisms of resistance. And what we did was to profile what would be possible susceptibility genes or S genes or genes that would be helping Striga to overcome pump-triggered immunity. So we have about 2 or 3 that we have identified. We have not validated them, and these are involved in the post-attachment resistance. So typical S genes that would be involved in the interactions between Striga and sorghum as helpers of Striga in the parasitism.
Okay. I also have a question from Tekle Haymanot, I think Tekle from Ethiopia, actually. Thank you, Steve, for the nice presentation. There is an ongoing work on the perennial rice by the Chinese Academy of Agricultural Science. Do you think Striga might remain a challenge in the case of perennial rice, for example?
I mean, perennial rice is good, and I think I'm familiar with that work. But being perennial does not stop Striga infestation. It will still be there, but maybe if you, it has nothing to do with, it will not reduce Striga infestation. Maybe less if you combine or intercrop and then grow it perennially, but in itself, perennial rice has little to do with Striga, little to do. But also I'm familiar with, I think it's the same group. It's the same group that developed Striga resistant sorghum based on strigolactone transporters. So if it is perennial rice with strigolactone, less strigolactone exudation, then maybe that's better. Or that would
Yeah,
work.
I wonder if in that case, if you have perennial rice, is Striga or these parasitic weeds, are they more seasonal or are they like they just when they find a host they germinate, right?
Yeah. So if you have perennial rice and if
You
you
will
plant
have
it, you still have striga in the soil, they will come out whenever rice is present. They will keep sprouting and sprouting. If you weed, then maybe that's fine and you don't have to plow. Maybe that will reduce. In that sense, maybe perennial rice would work better. In this case, you're not replanting and you're not allowing striga to flower. Maybe you can continue depleting the seed bank and you'll have a harvest in that case, or in that sense, then perennial rice would work. But it doesn't, it won't stop striga from germinating. As long as there's striga infestation in the soil, it will keep germinating. But if you're able to exhaust striga and if the perennial rice is able to withstand striga infestation this whole time, then you'll have better returns that way.
I do have some other questions, two questions from Chair Team now.
Good to see you, Tech.
And so this one person is asking, is the mutant host produced is resistant to all Striga species? So basically the lines that you're having, are they resistant to all the Striga species or only to this one? But I think you have mainly answered this already.
Yeah. Species, yes, at the level of species should work, and in principle should work for most variants of Striga. Even though we have, if the question that Shachien is asking is on the specific variants of Striga hamontica, it should be in principle, but we have not tested this. It's also possible that some Striga variants have receptors that can detect other strigolactone. So the strigolactone that is not active in the case of Robancol or in the case of the Striga smart sorghum. It's still possible that other Striga variants would have other sensing mechanisms, but in principle, it should work.
And to follow up on that, his second question is basically, can you have like infestation of different Striga species on the same host? So like multiple infestation, let's say?
Very unlikely. The ranges for Striga species are quite different. The natural ranges for them are different. So like in Kenya, we only have Striga hamontica in Western Kenya and some Striga asiatica at the coast. So those ranges rarely overlap. In Madagascar, you mostly have Striga asiatica. Yeah, so the ranges, it's unlikely that you'd have an overlap between Striga hamontica and Striga asiatica.
I have a question still coming up from Jacinta. In rice, is the Striga resistance not compromising the aroma trait?
We don't know. Yeah, I think that's a good question. We have not tested the aroma trait. I don't expect it to. I think there's already well-known data on using phytoalexins in rice. I don't know if they tested for aroma or other pleiotropic effects. For now, I think, like I said, we just want to test the hypothesis that this is the case, and then we'll jump off that bridge when we get to other things. For now, we just want to, but I think that's something that we should test. But I don't expect it to, because I've not read about that as a pleiotropic effect. As I said, they're already tested by Ruben's group at Iowa State for Magnapothe oryza. Maybe not. We don't know yet.
Okay. Thank you. I don't see any other question in the chat. I don't know if anyone still has a question, then it's time. Please raise your hand. If not, let me already thank the two speakers, Laurens and Steven, for a great presentation. I think it has raised some nice questions. So basically, I think it was very successful. In addition, we have had like around 50 attendees to this webinar. So I think that's quite a nice audience and coming from several countries in Europe, in Africa, and beyond. So thank you for your presentations. Thank you for answering the questions. Thank you to all the attendees for this online seminar, and we are looking forward to the next talk and see very nice publications on Striga, on plant transformation, and so on. Thank you very much, everybody.
Thank you, Mark.
Thank you.